I will start in a few minutes. Right, so I'll start the class by sharing my slides. This is being recorded as always. Okay, so this is the fourth live session of this course, NMR spectroscopy for structural biology. So I'll first go through the videos of week 4 and then I'll discuss some sample problems. So in the previous class, we had just started with some of the concepts of 2D NMR. This class, I will begin by introducing some terms. So in the 2D NMR, there are multiple pulses, and between each pulse, there is a certain time. And the magnetization evolves according to the pulse and the time. That is called the flow of magnetization. So magnetization flow is basically the way in which the magnetization is evolving because of the perturbation caused by the pulse and also the time that is allowed for it to evolve. So first thing is that we assume that all our spins are spin half. Now there are certain terms which I will define. One is IKZ. This refers to magnetization in the Z direction of spin K. So as you know, Spin K will have two energy levels, alpha and beta. And depending upon the population difference, N is alpha minus beta. So imagining N is the population difference of alpha and beta. Depending on this population difference, you will have the intensity of the magnetization. And here the magnetization is in Z direction, which is normally the direction of the unperturbed magnetization. So the term IKZ refers to the magnetization of the spin K in the direction Z. Similarly, if we have IKX or IKY, they are the X and Y magnetizations of spin K, which is basically the transverse magnetization. So you know that magnetization is initially in the Z direction, and then after a 90-degree pulse, it comes in the X or Y direction, where there is a detector, and the detector measures the FID. So the IKX or IKY is the one that is being measured. So if the detector is along the x-axis, the IKX will appear after. So the magnetization in the x direction will appear as an FID. And if you do Fourier transform of this FID, just one second. Excuse me 1 second. Right. So as I was saying, IKX is the magnetization along the x direction. Now, this will appear as an FID, and when you do Fourier transform of the FID, you will get signals. So the FID we saw in the previous class is basically with respect to time in the x-axis, and after you do Fourier transform, you will get frequency in the x-axis. So this is assuming the detector is in x-axis, then the signal along x-axis will be absorptive and it will be in phase. So both are positive direction and absorptive. But if the detector is along X, how will the signal along Y appear as? So the signal along Y, after you collect the FID and do the Fourier transform, it will appear as a dispersive signal. If you remember, dispersive signals basically have both positive and negative components. So this is a dispersive signal, and we are calling it in-phase dispersive because it has, the order is same, positive, negative, again positive, negative. So the order is same, so it is in-phase dispersive. Now these are magnetization of only one spin, that is spin K. Now what if you have spin K coupling with some other spin L? Then there are some terms that come. These are the terms which determine the what happens when two spins interact is depicted by these terms. So basically, if you have a term called 2IKX into ILZ, this means K spin has X magnetization and L spin has Z magnetization. And if you get the FID and do the Fourier transform, you will get anti-phase signals for these. So these are basically, we won't go into the mathematics of how these terms come, but all you can remember is if the magnetization of one spin is in X and in another spin it is in Z, that is, it's 90-degree to each other, then the signals will be anti-phase. Similarly, you can have the opposite. So here K had X and L had Z. Similarly, K can have Z and L can have X. Or one can have Y, one can have Z. So similarly, whenever you have two spins interacting with each other and one of them, so suppose one of them has Z magnetization and it has partially transferred some of its magnetization to another spin, which now has transverse magnetization. If that is the case, then you will get this kind of anti-phase signals. Now we will talk about some more terms. So in a two-spin system, we know that the energy levels both can be in the same direction as the external magnetic field. So the external magnetic field is in this direction. I'll call this B0. Both the spins can be up, that is alpha alpha. One can be up and down, that is alpha beta, or down and up, that is beta alpha. These two are degenerate, and both can be down, that is beta beta. So these are the four energy levels of a two spin system. Now we saw that the selection rule for transitions is that, so how do you calculate the M of a two spin system is you add up the individual M's. M of alpha is half, so half plus half is 1, and alpha beta have M plus half and minus half, so they cancel out and M becomes 0, and beta beta will have M of minus half, that is minus 1. The selection rule is delta M is plus minus 1. This is called single quantum transition or coherence. So the delta M can change only by 1. So the allowed transitions are shown here in the red arrows, where M can go from 1 to 0, or it can go from 0 to minus 1. So there are only 4 allowed transitions shown by the 4 red arrows. Now, what will the other transitions be called? So suppose you have a transition from alpha alpha to beta beta. The change in M is 2 because M is going from 1 to minus 1. That is called a double quantum coherence or DQ. And similarly, you can have a transition between alpha beta and beta alpha. So the M is not changing. So the delta M is 0, and that is called zero quantum coherence or ZQ. So in general, anything other than the single quantum coherence will be called as the multiple quantum coherence or MQ. Just 1 second, I'm having some network problems. Just give me 1 minute. Right. So double quantum coherence and zero quantum coherence are not directly measurable by the detector. The detector can basically only measure single quantum coherence. But just because you're not able to detect them does not mean that they are not appearing. So they are in the course of your magnetization, in the flow of magnetization, all these terms are appearing, but you are only detecting the red arrows shown. Now we learned about this kind of terms, which are basically terms made up of two spins, each of the spins having magnetization in a different direction, which is, so if one spin has magnetization in X, the other spin will have magnetization in another axis, 90 degree to X, so either Y or Z. So we were looking at these terms in the previous slide. These terms basically indicate a mixture of double quantum and zero quantum coherence. So you can add or subtract these terms in order to get pure double quantum or pure zero quantum coherence. I'll show you an example. Suppose, just suppose, this term is 2dq plus zq. Just imagine. And this term is just dq plus zq. So then if I'm calling this term as A, I'll call this term as B. So then if you do A minus B, you will get dq, which is pure dq. So that is what this says, that since these double intensity terms are mixture of dq and zq, you can add and subtract, basically linear combination of these terms will give you pure dq or pure zq. In this case, you can see the formula for pure dq and pure zq is written. So you have a flow of magnetization. So we saw that the flow of magnetization is basically the entire pulse sequence and all the time intervals. During this entire process, you can have you can have pure Z magnetization. You can have single quantum, double quantum, zero quantum, mixture of zero quantum and double quantum. You can have many different kinds of transitions. But the only transitions that your detector can detect are the single quantum transitions, which are shown by the red arrow. This is another term that I will talk about, which is when, so far the terms with two intensities that I was talking about had the intensities in two different axes, either x, y, or y, z. What if the intensity are both along the same axis? So 2ikz into ilz. So both the k spin and the l spin has intensity in the z direction. Then this is called two spin ZZ order. And this again can be converted into different terms. We won't go into the mathematics of that. So this is all about two spin system. Now you can have three spin system. Now, three spin system, again, you can have individual spins. So in the three spin system, suppose you have three spins K, L, and M. So IKX is X magnetization of the spin K. Similarly, ILX or IMX is talking about just the X magnetization of that particular spin. So you can see here, if I have, imagine the three spins are in a triangular arrangement. In this case, K will be interacting with both L and M. So IKX, if you take the FID and do Fourier transform, K will basically be split. So if you have K will first be split by L. So this one is JKL. And then each of these will further be split into JKM. So you will have a doublet of a doublet. That is what is shown here. Doublet of a doublet. The two spin terms are similar to what is discussed previously. You will have 2ikx, ilz. So there is a spin k in the x direction and L in the z direction. You can see the two axes are at 90 degrees to each other. Previously, if you did the Fourier transform of such a two intensity term, you will get one antiphase signal. So two signals antiphase with each other. Now you have three spins. So you will get another additional signal. So you can see that if it was only two spins, you would only get this part. Three spins will give you another additional part, and each signal is antiphase with each other. Then you will have your three spin terms. So you will have terms which include K, L, and M, all of them. And here the magnetization can be, one can be X, other can be Y and Z, two can be Z, one can be X. You can have many different kinds of the magnetizations. So basically the intensities of the different, each of the spins will have magnetization in a different direction. And you can combine these magnetizations to get different terms. In this case that is shown here, so let us look at the first case. The first case is 4IKX, ILY, IMZ. So K is in X, L is in Y, and M is in Z. This term basically gives rise to the summation of double quantum and zero quantum transitions of the K and L spin antiphase with respect to the M spin. So this term is something, it will form while the magnetization is transferring. This term can form, but you will not detect it because it is not single quantum coherence. Another term is the fourth IKX, ILZ, IMZ. In this case, if you do the take the FID and do the Fourier transformation, basically we will look at this in this way. We will imagine this term is 2IKX, ILZ, and another is 2IKX, IMZ. So this term is made up of these two intensity terms. And we know if you have a two intensity term like this, you will get an antiphase signal. Since there are two intensity terms, you will get two antiphase signals and they will appear like this. So you can imagine that. So if the Fourier transform, you are getting two antiphase signals due to each antiphase signal is due to one of these two intensity terms. And if you have if you have this kind of a term where there is nothing in the z-axis, so basically if you have this term where you can see K and M are in the x-axis and L is in the y-axis, so nothing is in the z-axis, that is called triple quantum coherence or TQ. So basically, again, we don't need to go into the mathematics or how these terms came or anything. Here, I'm just introducing you to the nomenclature. So basically, what does two spin system, what kind of terms they can have, what kind of quantum coherences they can have. So this is basically introducing you to the terms. We are right now not going that much into depth in the mathematics as to where the terms come from. So this is about the three spin systems. So now we'll go into something which is more practical, as in which you can visualize. We will talk about 2D correlation spectroscopy. Now, or it is also called COSY. Now, in 2D correlation spectroscopy, the pulse sequence is this. So you have the initial 90-degree pulse, which is the same in all the pulse sequences we have seen so far. Then you have a time t1, which is allowed for the evolution of the pulse. So the magnetization, the initial 90-degree term will flip the magnetization from the Z to the transverse axis. You can see initial magnetization of the K spin was in the Z direction. After the 90-degree pulse, it is coming to the transverse axis. Here we have considered Y. So IKZ is becoming IKY. Then you're allowing a time T1 in which the magnetization is evolving. So basically, we know by evolution of magnetization, we mean that the magnetization is here, it is rotating in the transverse plane. So we have given a time T1 and the magnetization will rotate with a omega K. Then we are giving another pulse and so this is the second pulse we are giving, and the flip angle of this pulse can vary. Here we have shown it as beta. And so when we are giving this second pulse, there will be mixing. What do we mean by mixing? By mixing, it means that the K spin will transfer some of its magnetization to another spin. Here we are calling it the L spin. So basically the purpose of the second pulse is that it is allowing the K spin to transfer some of its magnetization to the nearest neighbor spins. Here we are calling it the L spin. And then of course you have the time T2 where you are detecting both the K and L magnetizations, and you are getting a 2D spectra. So the time T2 is for the detection, and you will have magnetization of K, which was initially there, and the magnetization that K has transferred to L, which is also there. And each of these spins, so basically T2 will detect both these magnetizations and you will get this kind of a 2D spectrum. Here we are assuming a two-spin system. So we are assuming a two-spin system, AB. We know that in 2D spectra, basically both the axes have frequency omega 1 and omega 2. So if you only look at one axis, that is your normal 1D spectra. So A has a certain signal, B has a certain signal. Again, if you look along the other axis, A has a certain signal, B has a certain signal. Now, basically, diagonal peaks are, you can see this diagonal line that is shown. Whatever peak is on the diagonal line is the same in both the X and Y axis. You can see this small peak, which is the peak for A. So the peak for A, if you extend the peak of A both downwards and sideways, you will get the diagonal peak. And if you extend the peak for B, again downwards and sideways, you will get the diagonal peak. So the diagonal peak is basically the peak that is already there in the 1D spectrum. So it is not of much use. However, if you look at the cross peak or the correlation peak, which is basically also called the non-diagonal peaks. So the non-diagonal peaks, also called the cross peaks, are where all your information is there. So this peak, you draw a horizontal and a vertical to this peak. You can see that this peak is made up of the cross section, wherever A and B is meeting, that is where this peak is formed. So the information that this gives you, that A and B are correlated. Again, you can see the other cross peak is between B and A. So this cross peak is A to B. The other cross peak is B to A. So this, it is depicting transfer of magnetization from one spin to another spin. And in this case, it is telling you about which spins are directly correlated with each other. now we will look at how 2D correlation spectroscopy, or COSY, is allowing the magnetization to transfer due to J coupling. So here you have structure of a peptide. Peptide is basically NH, C alpha, CO. And you have the peptide side chain. In this case, it is a valine. And so the valine side chain is an isopropyl group. In this case, basically, first we know that there is a 3-bond coupling from NH to C alpha H. So here, between these two hydrogens, there is a 3-bond coupling. 1, 2, 3, 3 bonds. This we know this kind of coupling, we can call it 3J. So this will produce one. So wherever there is this kind of coupling, that means these two hydrogens are correlated and you will get a peak in the COSY spectra. So here, basically, the 1D spectra is not shown, but so for simplicity, the 1D spectra is not shown. So actually, there is one peak here, which if you come down to the diagonal, that is the NH peak. So instead of showing the 1D spectra, they have simplified it and shown the diagonal peaks, because we just saw that diagonal peak is the same as the 1D spectra. The NH and the C alpha H are correlated. So if you draw a perpendicular from the NH and the C alpha H, you will get a cross peak, which is because of this 3J coupling. The other, so this is from NH to H alpha. If the mirrored image of this, so if you imagine the diagonal is a mirror, this is your mirror image. This is coupling in the so suppose the cross peak I have circled is from NH to H alpha, then the opposite H alpha to NH will be the mirror peak. So we can only look at peaks on one side of the diagonal, that is enough. The other cross peaks, basically H alpha will couple to H beta, because this is also a 3J coupling. So H alpha and H beta will give another H alpha and H beta. They will give another cross peak. Then what will be the other cross peaks? Other will be H beta to gamma and H beta to gamma. So there are two kinds of gamma hydrogens. They come at slightly different chemical shifts, you can see, and H beta is showing correlation with both. So like this, in the COSY, the cross-diagonal peaks will tell you about which hydrogens are correlated by 3J coupling. So basically, the COSY will help in identification of the residue. Each amino acid will have a different side chain, and so each amino acid will have a different pattern of COSY. And so looking at the COSY pattern, you can predict that what is the amino acid that is present, and like that the sequence of the protein can be derived. Now there are some things to note. You can note that there is no 4-bond coupling. So you can by 4-bond coupling, suppose alpha to gamma. Alpha to gamma would be 1, 2, 3, and then 4. So there is no alpha to gamma coupling. So 4-bond coupling is not present. So this you can remember, that 4-bond coupling is too small that it cannot be observed in the COSY. And secondly, all the COSY cross peaks are within the valine. So the valine residue is basically starting from NH up to CO. This entire thing is valine. But So all your cozy peaks are within the valine residue. There is no cozy peak from the valine to the residue before it or the residue after it. So because of that, cozy is only useful in identifying that particular amino acid. But there is no transfer of the magnetization from one residue to the next residue or to the previous residue in cozy. For that, we will need a different kind of 2D spectrum. Now we will look at some more examples of cozy. So here, if you have a compound, and this is also a two-spin system, because you have HA and HX separated by 3 bonds, 1, 2, and 3. So there is 3J coupling between A and X. There is 3J, sorry, there's 3J coupling between A and X. So basically both A and X will appear as doublets. Imagine this is A, this is X. That is what is shown here. Both A and X are appearing as doublets. Now, suppose we are looking at the interaction between A and X. So in the 2D spectra, here we were just assuming that the cross peak is one circle. There is no, we are not, so far we have not looked into more detailed structure or fine structure of the cross spin. That is what we will do here. So basically here we have taken one cross peak and expanded it. And we have done the same for this cross peak. So we have the two cross peaks shown within the um, red circle are the ones we have expanded here. And now we will look at them. So basically, imagine that this is A, this is X. Again, this is X, this is A. So if you look at it that way, then this is your diagonal, and these are your diagonal peaks. We will, so on the left side, we have expanded one cross peak. We can see that one cross peak is actually, there are 4. Why are there 4 options? Because this is also a doublet, and this is also a doublet. So each doublet has 2. I'll just cross this out. It's becoming a bit crowded. So basically, from this doublet, there are 2 lines. And from this doublet also, there are 2 lines. So you are getting 4 peaks within the cross peak. So if you expand this cross peak, this is, if you are expanding the AX cross peak, you will get this kind of fine structure where there are 4 peaks. And if you expand the diagonal peak, so 1 second. Yeah. So, and if you, in this expansion, you're expanding the diagonal peak. The diagonal peak are all the same phase. So if you look at this diagram now, so what we have done here is that each, so here there are 4 peaks, and the 1D spectra of these have been shown. So how are we doing? First, let us understand the 2D spectra that is here below, and then we'll talk about the 1D spectrum. So once we have, this is the cross peak. If you expand the cross peak, you will get 4 smaller peaks inside the cross peak. And why you are getting 4 peaks? Because A is also a doublet and X is also a doublet. Now, if you look at, for now, you forget about the signs. I will talk about the signs when I come here. And if you expand the diagonal peak, there also you will get 4 peaks within the diagonal peak, because each is a doublet. You can see X is a, sorry. Yeah. So X is a doublet on both sides. And so if you expand the diagonal peak, you will again get 4 peaks within the diagonal peak. This is called fine structure. Now what has been done is that, so one line has been drawn through, you can see there's 1 peak here, 1 peak here, 1 peak here, 1 peak here. So there are 4 peaks in the horizontal line, and one horizontal line has been drawn through here. So that will only be talking about F2. So if you draw horizontal lines along each of these peaks, I'll draw this better. So you can see these blue lines that are there. So basically, imagine drawing a line like this blue line. So in the one, and this is you will, so if you draw one blue line and you see it in the 1D spectra, 4 peaks you will see, because there are 1, 2, 3, 4 points that the blue line passes through. So like this, if you look at this first blue line, the first 2 signals will come as antiphase absorptive. So you know this kind of signal is an absorptive signal, and they are antiphase because they are in opposite directions. And if you look at the other 2 dots here, they will come as dispersive in phase. Similarly, if you look at the second blue line, the first 2 dots will come as, again, absorptive antiphase, but in the first one it was negative positive, this is positive negative. And these second 2 will come as dispersive in phase. So now, if you look at these 2, is basically for this. So now if you write down the symbols are minus, plus, plus, minus. That is what is written here. Minus, plus, plus, minus. And for the second red circle, it is this. You can see all are positive. All are positive. So one thing to remember is that diagonal peaks are basically peaks along this diagonal. Diagonal peaks are always in phase, so all are positive and they are dispersive. And cross peaks are always antiphase, so minus, plus, plus, minus, antiphase, and they are absorptive. Now the separation between the, so this is called the fine structure. So there is 1 peak here, which if you resolve it further and look at the fine structure, you will see 4 more peaks. Now, if you look at the separation between these 2 peaks or between these 2 peaks, they will give you the J value, because we know that this separation is the J value, and that is what this separation is giving you. Or we know, if we look at the vertical axis, that this separation is the J value. That is what this is giving you. And we know AX and JXA is the same. So you can calculate either of these separations to get the J value. But it is better to calculate the J value from the absorptive spectra, that is the cross peak, because you can see in the absorptive spectra, just 1 second, I'll discard this now. So you can see in the absorptive spectra, the circles are very distinct. So you can clearly calculate the J value from the midpoint of one peak to the midpoint of the other peak. But in the dispersive spectra, the line shape is not good. You are getting a very spread out signal. So J value cannot be calculated from the dispersive signal. J value cannot be calculated from the dispersive signal, that is the diagonal peak. Now, the coupling constant, here you can see this red arrow, it is giving you the J. But if the J is very small, the fine structured peaks will merge with each other. And so here clearly there are 4 peaks within the cross peak. But if the J value is very small, you will not be able to distinguish the 4 peaks, and they will appear as one peak like this. That is one of the, we saw here that 4 bond coupling was not seen. If you see this point, there was no 4 bond coupling. So why 4 bond coupling is not seen is we know that 4 bond coupling is very small. It's hardly 2 to 3 Hertz. And because it is so small, these peaks will merge with each other. Now these peaks are anti-phase. So if they merge with each other, they will cancel. Minus and plus will cancel, plus and minus will cancel. So altogether, your cross peak will fully disappear. So if your J value is very small, the fine structure will merge with each other. And because the fine structure peaks are of opposite signs, they will cancel and your cross peak will disappear. That is why 4 bond couplings are not seen, because the J value is very small. So this is all about COSY. Now there is a special kind of COSY called as double quantum filtered COSY, or DQF COSY. In this, the initial pulse sequence is the same as COSY. You have a 90 degree pulse, then a time t1 for evolution, then another 90-degree pulse. So up to this, it is the same as COSY. But the difference is that a small time delay delta of around 100 microsecond is given, and then a third 90-degree pulse is given, and then T2 is as normal. So this is where the difference is. In normal COSY, after the second 90-degree pulse, you will immediately collect the FID and do the Fourier transform. But in DQF COSY, you give an additional time delay delta of around 100 microseconds, and you give a third 90-degree pulse. After that only you collect the FID. So what is it that DQF COSY is doing? Basically, what happens is that normally after this 90-degree pulse, this second 90-degree pulse in the COSY causes mixing. That is what we saw. That is transfer of magnetization of one spin to the other. So in DQF COSY, basically, we manipulate the mixing and the transfer pathway so that after the second 90-degree pulse, only double quantum coherence is present. That is why it's called double quantum filtered COSY. because you are manipulating the mixing and this time delay in such a way that after the cozy sequence is completed, only double quantum coherence is present. So you're filtering out all other like zero quantum, single quantum, multiple quantum. You're filtering out all other quantum coherences and only double quantum coherence is present. But remember that double quantum coherence cannot be measured. So that is the purpose of this third 90-degree pulse. It is converting the double quantum coherence to single quantum coherence so that it can be measured. So what is the advantage of this DQF COSY? Firstly, the resolution is better. So you can see this comparison of COSY and DQF COSY. Along the diagonal, we saw previously also that the diagonal peak, you expand the diagonal peak and you can see the peaks are not very nice. They are spread out. They are not well resolved. So DQF COSY takes care of that. You can see the diagonal peaks which are so much spread out in COSY. In DQF COSY, they are becoming much better resolved. So how that is happening is here the diagonal peaks were dispersive in nature. And dispersive line phase gives, dispersive line shape gives rise to very spread out signals. Basically, the pulse sequence of DQF COSY converts the dispersive line phase to absorptive. So this portion becomes absorptive. So because it becomes absorptive, it will give sharp signals like this. And so along the diagonal, the resolution becomes better. So what is the advantage of this? So what they have done is that along the diagonal, they have expanded this portion. And similarly, they have expanded this portion. So suppose there are some cross peaks along the diagonal. After expanding, you can see that this is a cross peak. This is a cross peak. These are all near the diagonal. But in COSY, the diagonal peaks are so much spread out and they are not well resolved that all these cross peaks are getting lost. In DQF COSY, because the diagonal peaks are now becoming well resolved, you can distinguish the other cross peaks that are near the diagonal. So that is another advantage of DQF COSY. And secondly, there are singlets are eliminated. So what is singlet? Singlet is basically something, a proton that does not couple with any other. So suppose you have one proton here, which is not coupling with any other proton. So this proton will only give rise to a diagonal peak. And that is very useless. If there is a proton which is not coupling with any other proton, it will not generate any cross peak. So that proton will only appear in the diagonal peak and you don't need that because it will only cause a confusion. So it is always good to eliminate singlets so that the spectra is not that much crowded. The third advantage of DQF COSY is that all singlets are eliminated. So any proton that is not coupled with some other proton gets eliminated. An example of such a proton is basically, let me. So if you have an aldehyde proton, imagine, and then suppose you have other whatever CH2, CH2. This proton is a singlet because it is not coupling with any other proton. The aldehyde proton is not coupling with the other proton because there's a quaternary carbon here. It is not able to couple with the other protons. So rather what it will do is that it will produce a singlet, and if you do DQF COSY, that singlet will get eliminated. So DQF COSY is a way of refining the COSY so that you get better resolved spectra. So now we will talk about resolution in 2D spectra. So basically resolution, which we will call R, is directly proportional to 1 by the acquisition time. So in the F2 dimension, the F2 or the omega 2 dimension is basically where the FID is collected. Now we have learned this earlier as is that how the FID is collected is that multiple points are collected at certain time intervals, and when you like average out all these points, you get the FID. So if the number of points collected is n and the time interval between each point, so this time interval between each point is dw or dwell time, the multiplication of number of points into the dwell time will give you the total time required to collect the FID, that is the acquisition time. Resolution is inversely proportional to the acquisition time. So that is something you can remember. And so in the T2 dimension, resolution is easy because you can just increase the acquisition time and your resolution will improve. So like suppose just from 50 microsecond, if you make the acquisition time as 100 microseconds, the resolution time will improve by the resolution will improve 2 times. And so it is very easy to improve resolution in the F2 or omega 2 dimension. Now what about the F1 or omega 1 dimension? Now if we look back to what we saw in the previous class, basically where the F1 and F2 comes from is that and this time T1 is varied. Initially T1 is taken as 0, then it is increased by delta T1, 2 delta T1, and so T1 is incrementally increased, and each time after mixing, that is the second pulse, the T2 is collected. So for each value of T1, you are getting a different FID, and the 2D Fourier transform of all these FIDs are giving you the 2D spectrum. So if you have to, so in terms of T1, so we saw that acquisition time in case of the omega 2 or F2 dimension is very easy. You just have to increase the time in which you are collecting the FID. But if you want to increase the acquisition time of T1, basically T1 is n, that is how many, so suppose the increment, so here T1 is 0, here it is delta T1, here it is 2 delta T1, here it is 3 delta T1, and so on. So the increment is delta T1. Increment is basically, and n is how many FIDs we are collecting. In this picture, we have just shown 5 FIDs. So n is the number of FIDs we are collecting. So if we want to, so acquisition time in terms of T1 is the number of FIDs we are collecting into the increment time between each FID. So if we want to increase the acquisition time, so if we want to increase resolution along the F1 axis, we have to increase the acquisition time of T1. But in order to increase the acquisition time of T1, we basically have to collect more FIDs. And that is a very time consuming process because collecting an FID involves the preparation time, the evolution time, the mixing time. It involves the entire pulse sequence. So in order to improve resolution along F1, we will have to increase T1 acquisition. And in order to do that, we have to increase the number of FIDs we are collecting. And that is a very time consuming process. So that is the limiting factor. F2, we can very easily increase, that is in the terms of milliseconds, so that will not matter. But the entire process of collecting one FID is quite long. So if we are, suppose from 5 FIDs, we are increasing to 10 FIDs, then the entire experiment time you have to increase by that much. So that is a very long time. And so it is a limiting factor to increase F1. Typically, one does 512 increments of T1 in a COSY or DQF COSY experiment. If you need more resolution, more number can be done as always. So greater the number of FIDs you collect, greater will be the resolution. So far we have looked at the, so this we were calling as the fine structure. This is the fine structure of a two-spin system, HA and HX. That is a two-spin system. Now we will talk about the fine structure in a three-spin system. Just give me 2 minutes. Right. We will now talk about the cross-peak fine structure in a three-spin system. Now, first we will imagine a three-spin system that is linear. So A, M, X in a linear way. So in this case, we have shown A will interact with M and it will split up into a doublet. You can see the doublet here. Again, X will interact with M. So X will also split up into a doublet shown here. M, on the other hand, will interact with both A and X. So M will first split up into two parts because of interaction with A. And each of these parts will split up into two more parts because of interaction with X. So you will get a doublet of doublet, that is DD, in case of the M spin. Now, what will be the signs? So in case of A and X, you are only getting two. So right now we are, suppose we are considering, so how will the 2D spectra look like? We'll ignore the diagonal spins. We will have cross spins. So this cross spin, so this is the, you can see that A is this, M is this, and X is this. So A and M will interact. Again, M and X will interact. We'll ignore the mirror image cross spins. So now we will look at this particular spin, which is the interaction of A and M. So if you look at this, so we have A on the y-axis, M on the x-axis. A is a doublet, 2 peaks are shown. M is a doublet of doublet, 4 peaks are shown. In this case, we call AM as the active coupling. So the peak is directly forming from AM, peak is due to A and M. So JAM is the active coupling. And active coupling will always cause anti-phase splitting. So we know that A is only interacting with M. So A will split into a doublet because of the M. And we just saw JAM is active coupling. So it will be anti-phase. plus and minus. M again will first split into a doublet because of A, and JAM is the active coupling, so it will split up into plus and minus. After that, M will be split up by X into two more doublets. This splitting is called passive coupling because the cross peak is due to A and M coupling. So that is called direct coupling. All other couplings are the passive coupling. So MX is passive coupling, and in passive coupling there is no change of sign. So plus will remain plus and minus will remain minus. So now you have four peaks. So now you have four peaks in the x-axis due to M. That is plus plus minus minus. That is what is written, plus plus minus minus. And in the y-axis, you have two peaks due to A, that is plus minus plus minus. So now if you multiply the signs, so plus and plus will give plus, minus and plus will give minus, plus and minus will give minus, and minus will give plus. So this will be the signs, that is the phases of this cross peak fine structure. This is assuming AM is greater than MX. So you can see this splitting that we have assumed. AM is large and MX is small. That will give this kind of a fine structure. What if you have the opposite? AM is smaller than MX. In that case, first the M will split into a doublet because of A, and JAM is your active coupling, so it will give antiphase splitting plus and minus. Now we are splitting into MX, and the condition here is that MX is greater. So MX will split in this way that the two peaks are crossing each other. So the splitting here is such that it is crossing with the other splitting. So plus will split. So plus here will split into plus and plus, because in passive splitting, passive coupling, there is no sign change. And minus will split into minus and minus. So the order gets changed. So now this is the fine structure of M, which we are putting in the x-axis, plus minus plus minus. Fine structure of A remains same. And so now the fine structure gets changed. So depending on whether JAM is greater than MX or the opposite, the fine structure will be different. So now if we look at the mirror image cross peak, so we will now look at this one. In this cross peak, we had taken M in the x-axis and A in the y-axis. In the mirror image cross peak, this one, we will take the opposite. So we will take A in the x-axis is A, and we will take M in the y-axis here. So now if you look at the fine structure, it is different. So JAM greater than MX. So the first case. So you can see the fine structure is not the same if we take this as case 1 and case 2. So fine structure is not, so on the either side of the diagonal, the peaks are not symmetric. They have different fine structure. So this is all about the linear system, linear three-spin system. Three-spin system can also be triangular. So you have A, M, and X arranged in a triangular fashion. In this case, there are some additional peaks. So you have the peaks that you had earlier, A interacting with M and M interacting with X. But what are the additional peaks you have now? You have this peak, which is A interacting with X, because, and you also have, so X interact, so in this one, each A is interacting with M and X. So if you, I'll just escape this. So A is now not only interacting with M, but A is also interacting with X because there is a triangular orientation. Now, in this case, each A, M, and X, all of them will have doublet of doublet because all of them are split up by two other spins. Now we will concentrate on this spin, which is shown in green here. So if we are looking at this spin shown in green, that is, we are looking at the mx. So m in the y-axis, x in the x-axis. So initially, x is being split up by m, and that is your active coupling, and it will give rise to antiphase. So it will become plus and minus. So x has already been split by m. Now x will be split by a. That is your passive coupling, and the sign will not change. So plus will give plus and minus will give minus. Similarly, M is initially split by X, which is your active coupling, because we are looking at the MX peak. So there's plus minus, and then M will be split by A, which is your passive coupling. So the sign remains unchanged. Plus remains plus, minus remains minus. So now in the y-axis, we are writing M. Fine structure of M is plus plus minus minus. We are writing plus plus minus minus. In the x-axis, we are writing x plus plus minus minus plus plus minus minus. Similarly, we are multiplying. So now we will have 4 into 4, 16 components. So the fine structure of each cross peak will have 16 components. So that is about the, in the previous case, you had 4 into 2. So you had 8 components in the fine structure. Now you have 16 components in the fine structure. So the fine structure depends on the active and passive couplings. It will also depend on the relative magnitude of the active and passive coupling that we saw in the previous slide. So the relative magnitude, whether the active coupling is larger or the passive coupling is larger, the fine structure will be different. And finally, the peak appearance on the two sides of the diagonal will be different, which we already saw. This is the peak appearance when you have M in the y-axis and A in the x-axis. And this is your peak appearance in the opposite side of the diagonal. And they are clearly different. So this is all about your three-spin system. Now we'll look at a special kind of cozy known as the J-scaled cozy. So in J-scaled cozy, what happens is that you have the initial 90-degree pulse and you have T1 that was already there in normal cozy. But in normal cozy, after this T1 time, you are giving the second 90-degree pulse. Here you're introducing additional time delays of tau. So, and then you're introducing another 180 pulse, again another time delay of tau, and then a 90 pulse, and finally you are recording it. So, additional time delays of tau are introduced. So, one condition that is maintained in this J-scaled cozy is that the time between the first 90 pulse and the final 90 pulse is your alpha T1, where alpha is greater than 1. So if alpha is greater than 1, alpha T1 is greater than T1. So basically, the initially in cozy, a time of T1 was given. Now additional time delays of tau are added so that the total time from the beginning to the up to the collection is alpha T1. So first of all, why are we doing this? The thing is that in order to prevent loss in intensity due to cancellation of antiphase signals, our strategy is to scale up the coupling constant along the F1 dimension by designing a suitable pulse sequence. So we already saw that we were seeing here that if the J value is small, the antiphase signal will cancel out and you will lose the cross peak. So we have to increase the J value, that is increase the resolution. But increasing the resolution is easy along F2. So now we want to increase the J value or resolution along F1. And that is why this pulse sequence has been designed, which is the J-scaled COSY, so that we can increase the increase the J value, that is the resolution along F1, and that will help us to get better resolved cross peaks. And we will not, the problem that was happening, that cross peaks were getting cancelled out because the antiphase signals were merging, that will not happen. So basically, we know that so up to this point, 90 T1. So we are applying a pulse of 90 and we are allowing the magnetization to evolve for time T1. So in this time T1, your chemical shift is evolving. But after that time T1, you can see this tau 180 tau. This is the sequence of your spin echo. And we know that spin echo will refocus the chemical shift. So chemical shift evolution is only happening in time T1. Because for the rest of the time, that is tau 180 tau, the chemical shift is just getting refocused again. So time of evolution of the chemical shift is T1, but time of evolution of the J value is the entire time alpha T1. Because we know that spin echo does not refocus the J value. So spin echo will not affect the J value and the J value will continue to evolve for the entire time period alpha T1. So now an example is shown here. Basically, this is uracil and this is also a two-spin system where there's 3J coupling between H5 and H6. So now let us look at the cross peak. In this cross peak, omega 2 is 7.6 Hertz, which is the J value. And omega 1, if you see, it is 4 times the J value. So this distance is the J value along omega 2 and this distance is the J value along omega 1. J value of omega 1 has become 4 times the J value of omega 2. So we are scaling up the J value along omega 1. That is, we are increasing the resolution along the omega 1 or F1 axis. So this is the result of this kind of J-scaled COSY. And this higher resolution along omega 1 or F1 is important because, as we discussed, that is the limiting step. It is easy to increase resolution along F2, but it is difficult to increase resolution along F1. So by doing the J-scaled COSY, the resolution along F1 is able to be increased. That is, the J value along F1 or omega 1 is able to be scaled down. And that is the advantage of J-scaled COSY. Now there is another kind of COSY called omega 1 decoupled COSY or CT COSY, where CT stands for constant time. Here the pulse sequence is similar to the J-scaled COSY, except here it was alpha T1. So with the variation of T1, the alpha T1 was changing, and so you could collect multiple FIDs and get the 2D spectra. But in this case, the total time from the first 90 to the final 90 degree is a constant time period delta. This is the time period that was varying in J-scaled COSY. In J-scaled COSY, this was alpha T1. So it was variable. But now this time period is constant. So if this time period is constant, what will happen? If I increase T1, tau will reduce so that the total time is maintained as constant. So the consequence is that, so we know that J evolution occurred for the entire time period from beginning to end. Now, because the time period from 90 to the last 90 is constant, the J evolution is also constant. So J coupling will not appear along omega 1 axis because there is no evolution of J. So if you remember earlier, we used to have so in the cozy, if you can see in the cozy, you have in the y-axis also you have delta plus J. In the x-axis also you have delta plus J in the cozy. In CT cozy, what you are doing is you're making the total time from the first 90 to the final 90 constant. If that is constant, so this is the time period T1. That is, it will affect the axis omega 1. So in omega 1, this total time is constant. If the time is constant, there is no J evolution. And so the J information will disappear from the omega 1 axis. You will only have delta along omega 1 and omega 2 remains same. It will be delta plus J. So you are losing the J information from the omega 1 axis because you are not allowing J to evolve. Since the total time is constant, J evolution is also, there is no J evolution, and so the J information disappears from the omega 1 axis. So we can imagine that there is decoupling along the omega 1 axis. If there was coupling, we would see this kind of splitting. Decoupling means the splitting disappears and we will get this kind of peak. But we are not losing any information because your omega 2 axis is still having the J information. So we are not losing any information. What is the advantage of this? Suppose you have two cross peaks like this. So this is one cross peak, this is another cross peak, and they are overlapping. So this kind of overlap can cause cancellation. But if you do a omega 1 decoupled cozy or a CT cozy, then the spectra will become less crowded, so there will be no cancellation. You can still get the delta information. So suppose you draw here, you will get the delta information and the J information you will get from your omega 2 axis. So no information is lost. So CT cozy is a way in which you can make the spectra less crowded and you can avoid cancellation of intensities. Now we will talk about another experiment, which is called a total correlation spectroscopy or TOCSY. What TOCSY does, it will completely eliminate any antiphase character in the peaks and you will only have in-phase character. So we won't bother about the pulse sequence. Basically, here a comparative spectra is shown between COSY and TOCSY. COSY will only show cross peaks between the immediate neighbor. So A will show with M, M will show with both A and X, X will show with M and Y, and so on. But TOCSY will show all possible correlations. So A will go with M, A will go with X, A with Y. So in TOCSY, the number of correlations is much larger. So you can see in COSY, the network of coupling is only between the nearby neighbors. In TOCSY, first of all, you have cross peaks with neighbors that are much farther away from the which are separated by 2 bonds, 3 bonds, and even more number of bonds. So in Toxi, you can already see the number of peaks is much higher. Secondly, in Toxi, the plus minus or antiphase character is completely eliminated. So all the peaks will have in-phase character, and so cancellation effects are eliminated. In COSY, you know that if there is overlap of peaks due to antiphase character, some peaks can get cancelled out. But in Toxi, this cancellation effect is eliminated because all the peaks are in phase and the number of peaks are much higher. So you can see resolution in Toxi is much better because there is no cancellation of peaks. So another advantage of Toxi is that suppose you have three protons A, M, and X, and you have some other proton M dash which is overlapping with M, and you are getting this kind of a Toxi spectra. So you have a AMX connected system and you have some other proton M prime which has the same chemical shift as N. So in COSY, you can see that A is interacting with M, M is interacting with X. But we cannot predict whether the system is AMX or the system is AM plus M prime X. Because if it is AMX, then also A and M cross peak will be there, M and X cross peak will be there. And if the system is AM and MX, A and M cross peak will be there. And because M dash and M are at the same chemical shift, the M dash X cross peak will also be there. So COSY cannot differentiate between these two. But suppose I take the Toxi and I get this kind of a spectrum. So Toxi is showing A to M cross peak. It is also showing A to X cross peak. So this will now prove that the system is AMX, because only when it is a connected system like this, Toxi will show correlation of A with both M and X. So Toxi will help to differentiate between these two systems. So there is this kind of a relay of magnetization in Toxi, which helps us understand the connectivity in the system. We will talk about another kind of spectroscope. Just 1 second. We will talk about another kind of spectroscopy, and this is called XC, that is exchange spectroscopy, or NOESY, that is NOE correlated spectroscopy. So basically, XC is used in situations where there is a chemical exchange. So if you remember the dynamic NMR, HA and HB, they were exchanging with each other. That kind of situation is for XC. And NOESY is mostly used for structure determination in large biomolecules. We'll see how that happens. So what happens? The pulse sequence of these two are the same. You have an initial 90-degree pulse. So if you have IKZ, that is Z magnetization of spin K, after the initial 90-degree pulse, it will become transverse magnetization. So it will become IKY. Now, after a time T1, we will adjust the time T1 in such a way that after the time T1, we will apply the second 90-degree pulse and the magnetization becomes minus IKZ. So this is, we have to adjust it in such a way. There is a mathematical way to adjust it. We don't have to worry about that. Basically, we have to adjust T1 in such a way that when we apply the second 90-degree pulse, the K magnetization is completely in the minus Z axis. After that, we allow a certain time tau for mixing, that is for transfer of magnetization from K to L. So suppose a portion of the K magnetization is transferred to the L spin and it becomes ILZ. Finally, you apply a final 90-degree pulse and you collect the FID. So the FID will have components of K as well as L magnetization. Here you can see that the final 90 degree is basically transferring both K and L magnetizations to the transverse plane, that is Y, because the detector is in the transverse plane. So the detector is detecting IKY plus ILY. So the Y magnetization of both K and L. So this is the pulse sequence. Basically, in tau mixing is where the magnetization is getting transferred. And this happens when the two spins are close in space. It happens through dipolar interactions, which is the nosy, or by exchanging, which is the X. So that is the pulse sequence. So in the nosy, again, this same pulse sequence is shown. Basically, we saw this in the previous class when we were talking about NOE. There is something called the correlation time, which measures how fast the molecule is tumbling or moving inside the solution. For small molecules, tau c is of the order of 10 to the power minus 10 to 10 to the power minus 12 seconds. And for large molecules, tau c is larger because a large molecule will take longer time to move about. So it is of the order of 10 to the power minus 8 to 10 to the power minus 9 seconds. We also discussed that how NOE can be measured. We show it as NXA, where A is the perturbed spin and X is the spin to which the perturbation is relayed. So in the previous case, we were perturbing K and K was transferring magnetization to L. So K, so basically in the previous example, so ours will be, we were perturbing K, so it will be NKL. For small molecules, the NOE was half of the gyromagnetic ratios gamma A by gamma X. And for large molecules, it was minus the ratio of the gyromagnetic ratio gamma A by gamma X. So for large molecules, we already know that the NOE is negative. So for now, we will consider homonuclear system. So the gamma will cancel out. NOE will be minus 1. So NOE is negative for large molecules. That is, omega tau c is much greater than 1. We know tau c is large, so omega tau c is greater than 1. So, and in small molecules, indeed it is positive. And again, we can say the omega tau c is much less than 1 because omega tau c was small. So what happens to the NOE nosy spectra, which is NOE correlated spectroscopy? So because for large molecules, NOE is negative, both the cross and diagonal peaks will have the same sign. You can see diagonal peak and cross peaks are all having the same sign. On the other hand, in small molecules where NOE is positive, the diagonal peaks are in the positive direction, cross peaks are in the negative direction. You cannot see the cross peak, they are below the plane. You can just imagine they are in the negative direction. The intensity of this cross peak in NOE is proportional to the mixing time. So more time you allow for mixing, more is the transfer of magnetization and greater will be the cross peak. Then it is the T1 or spin lattice relaxation time, and also the spectrometer frequency and also the exchange rates. So these are the factors on which the intensity of the NOE cross peak depends. NOSI basically shows your inter-residue correlations with the residue preceding it. So here again you have a peptide and you can see that the NOSI, so NOSI basically shows whichever protons are close in space, they, if the protons are close in space, they are close at for transfer of magnetization, that is dipolar coupling, and so they will show NOE cross peaks. The intensity of NOE, we have already seen, is inversely proportional to 1 by R to the power 6, where R is the distance between the two protons. So here some NOEs are shown. So alpha and NH of the same amino acid, they are close in space, they will show an NOE. NH will also show with beta and with the two gammas. So these red lines are all the NOEs that will be seen. And more importantly, the NH of one amino acid will show NOE with NH of the previous amino acid. So the problem of COSY we were discussing is COSY is only restricted to that particular amino acid residue. But NOE can show correlation with preceding amino acid residue, that is inter-residue correlations can be there. And if we have inter-residue correlations, we can find out the sequential connectivities using NOE. Now, some things to remember is that NOE can only be observed is that if the distance between the two protons is less than 5 angstrom. Because greater than 5 angstrom, this term becomes too small and you can't see the intensity is so low that you can't see it. Another thing is that it is not that you can infinitely keep decreasing R and get extremely high intensity. That is not possible because no two atoms can come closer than 2.4 to 2.5 angstrom due to steric clash or van der Waals clash of the atoms. So if your atoms are large, this distance will be larger. If the atoms are small, this distance will be smaller. So you cannot keep on decreasing R. R can only decrease up to a certain limit. So what do we mean when we say sequential connectivities? So let us imagine we are starting from NH of I plus 1. NH of I plus 1. Basically these light blue or cyan colored residues are the residue I or valine. And these dark blue residues are the residue I plus 1. So now we will only focus on the light blue and dark blue filled circles. We will not look at the unfilled circles right now. Still, I'll just say the unfilled dark blue circles, the hollow circles that are dark blue are the residues I plus first residue, and the hollow green circles are the I minus first residue. So if we start from NH I plus 1, I am moving up and we come here. This is NHI. So NHI plus 1 and NHI is showing connectivity. And NHI is also showing connectivity with this green circle, that is H alpha. This green circle is H alpha I minus 1. So that is not shown here, but after NH, H alpha will come. So basically, this NH is connected with the previous NH. The previous NH is connected with the previous H alpha and so on. So you can see a sequence. Similarly, if you start from NH to the beta, NH is connected to beta. NHI plus 1 is connected to beta of the I plus 1. So NHI plus 1 is connected to beta I plus 1, shown by the red arrow here. Beta I plus 1 is connected to beta I minus 1. So the beta of the previous residue. Then that will be connected to the NH of the previous residue. That will be connected to the beta of the residue before that. So like that, there are sequential connectivities and it will help you predict the amino acid sequence of the protein. Now here a 500 megahertz 2D noisy spectrum of a protein is shown. Imagine a polypeptide A, B, C, D. So this is a polypeptide chain where you are looking at 4 residues A, B, C, and D. Now, the polypeptide chain can fold in many different ways. One way is like this. Here, the two end residues A and D are coming close to each other. So if they are close to each other, they will show a nosy cross peak. So if A and D show a nosy cross peak, that means the polypeptide has folded like this. Now suppose the polypeptide is folding like this. So A, B are far, but C and D are close to each other. So you will see a CD cross peak in the nosy spectrum. Or if it is folding like this. So the middle residues are coming close to each other. So BC will show a nosy cross peak. So depending on what nosy cross peak is seen, you will be able to predict that how the polypeptide is forming. That is what is the 2D or 3D structure of the protein. So the nosy spectrum is kind of like the fingerprint of the structure of the molecule, because as the structure of the molecule is different, the nosy spectra also is different. Now, from the intensities in the nosy spectrum, the inter-atom distances can be calculated. If you know the intensity, you can calculate R. And then we can try to build a model of the protein which matches the R inter-atom distances that we have calculated. So that this is the basis of structure determination of the biological macromolecules. So basically, from nosy, we calculate the inter-atom distances. Then we build a model of the protein where the inter-atom distances are what we have calculated. And that is how structure prediction is done. And why nosy is so important in predicting the structure of biological macromolecules. This is another experiment. We will go into this very shortly because we don't have this experiment will be gone into more detail in the next week. So I will just introduce the experiment. So far, we have been only looking at homonuclear 2D spectra. Whether it was TOXY or COSY or NOSY or XY, everything was homonuclear. Now we will look at heteronuclear single quantum coherence or HSQC. So here the correlation is heteronuclear. It is between hydrogen and 13C or hydrogen and 15N. This is important because the proton spectra can become extremely crowded. We have seen, look at how crowded this spectra is. So because the proton spectra, there are so many protons in the molecule and the proton spectra can become so crowded, the heteronuclear spectra also helps us understand different correlations. Now, another problem is that when you have a homonuclear system, you have diagonal peaks, but the diagonal peaks don't give us any information. The diagonal peaks are very strong, but they are useless. So heteronuclear correlation will not have diagonal peaks. It will only have cross peaks. So that is another advantage of the heteronuclear spectrum. And finally, for one thing is that for the heteronuclear experiments, we know that the population of 13C and 15N is not that much. Mostly the stable nuclei that is highly populated is 12 carbon or 14 nitrogen, which is not NMR active. So in order to do these HSQC experiments, we need to radiolabel the proteins with the 13C or the 15N so that we can get high enough intensity of the peaks for the experiment. So we will go into the details later, but this is the very basics of the HSQC. So this basically covers all the points of week 3. I'll quickly go through some sample questions. So first question is asking, in jump return sequence for water suppression, to excite a spectral region of 2000 Hz away from water, what is the value of tau in millisecond? So we saw this in the previous class. I'll come to it. So basically in jump return sequence, what we were doing, so the magnetization was along Z. We were applying a 90-degree pulse to bring the magnetization of all the spins along Y. Then the carrier was on water, so the magnetization of water will not evolve. It will remain along y, but the magnetization of your other spins will evolve. Then you apply a 90 minus x pulse so that the water spin will go back to the z-axis. And if a Fourier transformation is done, because the detector is only along x and y, the detector will not detect the water peak. It will only observe the peaks of the samples. And so the water will be suppressed. This was the basis of the jump return sequence. And the basically, how did we set this time tau, which we are allowing for the spins to precess? The formula was that in a time tau, so suppose we want high intensity of this blue signal, that is sample 1. In a time tau, the angle moved by sample 1 will be 2 pi omega 1 tau. And if this angle is 90°, that is pi by 2, that will give you the maximum signal, because 90° will bring the angle from y to x, and your detector is along x, so the detector will capture the whole signal and the intensity will be maximum. So in time tau, the angle moved 2 pi omega tau. If it is 90, that is pi by 2, then your intensity will be maximum. So if you rearrange this, tau will be 1 by 4 omega I. This is the tau we should be setting. Here, the question says that the spectral omega I, so this omega, so in the x-axis you have frequency, omega is 2000 hertz away from water. So tau is 1 by 4 omega I, 1 by 4 into 2000. 2000 is already given in the question. So tau is coming out to be 0.125 milliseconds, which is your option B. So your correct answer is option B. Option B is your correct answer. Just give me 2 seconds. Next, the question is asking, what is the spin echo pulse sequence? So spin echo, if you remember, we have the magnetization along Z. We apply a pulse sequence of 90, which forces the magnetization to come along Y. A time tau is applied to allow the Y magnetization to evolve, and it comes somewhere in the XY plane. After that, a 180 pulse is applied, which flips the magnetization in this direction. So basically, the X component becomes negative and the Y component remains same. So the magnetization comes along this direction. And after that, again, the same time tau is given so that the magnetization moves here and refocuses along the Y direction, so that these two are the same. So the entire pulse sequence is 90 tau, 180 tau. So here the option is 90 tau, 180 tau is B. So your correct answer is B. Then the question is asking, the spin echo refocuses what? The spin echo refocuses. So we have seen that the spin echo is refocusing the chemical shift. So it will refocus all chemical. Here we are showing all chemical shifts by this arrow. So all chemical shifts will get refocused by the spin echo procedure. So A is your correct answer. The spin echo is refocusing all chemical shift. And so B is incorrect because it is not some chemical shift, it is all. Similarly, D is incorrect. But what about the coupling evolution? We know that so suppose A is split into two parts by another nucleus X. We have A1 and A2. So A1 and A2 were initially in the Z direction. After the 90-degree pulse, they come in the Y direction. Then you apply an interval of tau, and because the A1 and A2 frequencies are different, they are moving in different directions. After that, the 180 pulse is applied, and again a time period tau is given. And as we discussed in the previous class, the A1 and A2 do not refocus. If they refocus, then the the coupling evolution would have refocused. But because at the end of everything, at the end of the spin echo sequence, A1 is still traveling along this direction and A2 is still traveling along this direction. So your coupling evolution is not refocusing. So your option C is discarded, and that leaves you only with option A. Then you have this question. In spin echo, what happens to the field inhomogeneity? Basically, here the example is shown. So you have the field along the Z. After applying a 90-degree pulse, the magnetization comes along Y direction. Now, because of field inhomogeneities, some of the spins will move faster and some will move slower, and so the spins are splitting up into different magnetizations in different directions. Why this is happening? Because of the field inhomogeneity. Now you apply the 180-degree pulse, which will completely flip the magnetizations in. So they were pointing this direction, now they are pointing this direction, because the 180-degree pulse have flipped them. But the important thing to remember is that here they were moving outwards. So they were diverging. But now the spins are moving inward. So the direction of movement of the spins is same. It is always, you can see the direction I've seen, the arrows are in the same direction. But now they are converging because of the 180-degree flip. So they are converging and they will again refocus back into the same direction. So the splitting that was happening due to the field inhomogeneity are again getting refocused back. So field inhomogeneity is not refocused. This is wrong. Field inhomogeneity is refocused. This is correct. What about, it's not partially refocused, it's fully refocused. This is wrong. What about the option D? Now, what happens is that while all this is happening, you have to remember that the magnetization always wants to relax back to the Z-axis, which is the spin-spin relaxation. And this relaxation rate is e to the power minus 2 tau by T2, where T2 is your spin-spin relaxation or transverse relaxation rate, and tau is this time interval you are giving. So spin echo does affect the relaxation, because normally your relaxation rate is just T2. But in spin echo process, normally it comes to the Y and immediately goes back to the Z. But because of the spin echo process, the relaxation rate is also getting slowed down by this rate shown here. So spin echo does affect the relaxation rate, and so option D is also wrong. So your correct answer is option B, which is shown here. Then you have this question. In the Watergate scheme for water suppression, what happens? So basically, if you let's look at each of the points. The first thing is saying employs spin echo sequence along with selective pulse on water. So what is the Watergate sequence? Basically, you have the spin echo sequence 90 tau, 180 tau, that is there. But along with that, you are applying certain selective pulses only on water. So these rounded pulses are selective pulses applied only on water, and you also have certain field gradients. What does the first option say? It employs spin echo sequence along with selective pulses on water. That is true. You have the spin echo sequence 90 tau, 180 tau, and you have selective pulses on water. So option A is correct. Option B is saying field gradient have no effect on water signal. Now that is not true, because what happens is that because of this field gradient, there's a field inhomogeneity. We know that the total effect of this pulse on water is zero, because water is going through, you can see, water will initially come to the transverse phase like this, but after that it is facing minus 90 minus 90 minus x, 90 minus x. So 180 minus x and 180 plus x. So this entire pulse has no effect on water. But what does have an effect is that the field inhomogeneity is causing the water signals to completely spread out and become and continue dephasing till it becomes zero. So field gradients have no effect on water signal is wrong, because they are having an effect in which water is completely dephasing. Then you have sample signals are dephased. That is wrong. Sample signal is focus and water is dephased. So this is wrong. Sample signal is dephased is wrong. And both sample and water is dephased is wrong, because only sample is refocus and water is dephased. You can see sample is refocused, water is dephased. So this is also wrong. So the correct answer is option A. Then you have a question. A proton exchanges between two sites A and B. The proton chemical shift difference between two sites is 100 Hertz. The two lines will get broadened when? So we know about exchange spectroscopy. So what the question is telling you is that this difference, frequency difference between the two sites. So there's an exchange between A and B. There's an exchange between A and B. And the frequency difference between A and B is 1000 Hertz. The question is asking when the lines will get broadened. Remember, the question is not saying when the lines will merge. It is only saying when the lines will become broader. So let us look at the options. Chemical exchange has no role to play in line broadening. This is wrong, because we can clearly see because of the exchange, a broadening is happening, then the lines are merging. If exchange rate is smaller than 1000 Hertz, this is wrong. If exchange rate is smaller than 1000 Hertz, that is your slow exchange, lines are very sharp. There is no broadening. If exchange rate is much larger than 1000 Hertz, this is also wrong. Because if exchange rate is much larger than 1000 Hertz, it is fast exchange. The two peaks will merge. The question is asking when the two peaks will broaden, not when the two peaks will merge. So B is also wrong. So the correct answer is option C. When the exchange rate is close to 1000 Hertz, you can see coalescence. When the exchange rate is close to 1000 Hertz, the peaks are getting broadened. That is what the question is asking. So the correct answer is option C. And you have one question asking NOE arises due to what? So if we look at the NOE, let us look at the options. NOE arises due to J coupling. This is wrong, because NOE depends on distance between the spins. You are perturbing spin A, and from A, the perturbation can transfer to B or C, not through bond, but through space. So it is not dependent on J coupling. That is wrong. And we know NOE depends on the distance between the two spins. It is directly proportional to 1 by r to the power 6. So this is also wrong, because NOE is dependent upon the distance. And so NOE arises due to dipolar coupling. This is correct. Your correct answer is B. So as I said, the NOE is the transfer of magnetization through space, and this is called dipolar coupling. So your correct answer is option B. Then you have, just 1 second. Then you have that steady state NOE. This we just discussed. That steady state NOE, this formula we discussed in this class only. Clearly for large molecules, it is negative, and for small molecules, it is positive. So if it is negative in large molecules, this is wrong. And for small molecules, it is positive. So this is wrong. Clearly, it is dependent on the molecule size. This is wrong. So B is the correct answer. Now it is asking about selective population inversion. In selective population inversion, two-spin system, where AX, where A is proton and X is 13C, what do? Basically, we apply a selective pulse, and this selective pulse will increase the intensity of the signal which had low intensity. So we are applying the selective pulse to A1. We are applying the selective pulse to the high intensity signal, and we are increasing the intensity of the low intensity signal. So in 1H and 13C, the high intensity signal is 1H. We know 13C population is low intensity signal. So in this case, high intensity signal is A, and that is where we are applying pulse. So we are applying the inversion pulse to 1H, which is A, and so inversion pulse is applied on X is wrong. Inversion pulse is applied on, this is also wrong. Inversion pulse is applied only to the high intensity signal, which in this case is 1H, which is A. So inversion pulse is applied to A, but we are applying it only, we have A1 and A2. We are applying it to only one of the A's. So this is also wrong because it is saying both the A's. So the correct answer is option B. And finally, we have one question on inept, where inept, it is saying that in an inept experiment in an AX system where A is 1H and X is 13C, for this pulse sequence, the polarization transfer occurs from where to where. In inept, we know that what happens is that basically the polarization transferred in inept happens from A to X. That is, it is happening in this case from 1H to 13C. And if you match the pulse sequences, the pulse sequence that is given in inept, so the initial magnetized 90, the initial 90x pulse, whichever nuclei you are applying it to, the transfer is occurring from that nuclei to the other nuclei. You can see here, initially you are applying the 90x pulse to A, and the magnetization is transferring from A to X. Here you are applying the initially 90-degree pulse to A, so the transfer must occur from A to X, in this case, option D. So option D is the correct answer. That covers the things for so I've covered the lectures of week 4. I've also discussed some sample questions of week 3. And as always, I'll upload the slides as well as the recording, and you can come back to me with your doubts. Thank you.